Thanks for that very kind introduction, Michael, and I feel very honoured to be sharing the podium with such august speakers. I'm a clinical geneticist, so I'll just forewarn you that my presentation is very much slanted in that direction, but I hope that you will appreciate that this technology really has a place throughout medicine. So when we talk about genetic disorders, often people will wonder just how common or how rare they are, and because of the presumed rareness, will not necessarily think that they figure very highly in medical practice. But indeed, they affect more people than we realize, and these are just some statistics to put that into light for you so that you can appreciate that. When we think about diagnosis of genetic disorders, the traditional approach in the past has been a combination of an initial clinical evaluation followed by some general and then perhaps targeted biochemical screens. And then the diagnostic process is ramped up with a whole raft of additional investigations, including karyotype imaging, histopathology, enzymology. And then if you're, after all of that, there is, you have a very firm idea of what the disorder might be, then there might be one or a few genes that you can then potentially move on to and sequence in an effort to try and confirm your suspicion. And in reality, traditionally that has been potentially a very long and expensive and sometimes intrusive in terms of invasive procedures that need to be done, and ultimately may not lead to a genetic diagnosis throughout that odyssey. Enter the next generation sequencing era. So of course we've all heard of the Human Genome Project, and one of the major aims of the Human Genome Project was to sequence the human genome, and that actually was accomplished a couple of years ahead of schedule and came in slightly under budget at around 2.7 million US. One of the major outcomes of the Human Genome Project had been a steady improvement in sequencing technology and also the ability to process the massive reams of data that Roger alluded to, and so much so that the cost of genetic sequencing has plummeted over the last 5 or 10 years, and so much so that by early 2014, using Illumina's latest technology, and I'll just say here I have no stock in Illumina and I receive no funding support from Illumina. Their technology allowed whole genomes to be sequenced for around about $1,000 US. So a dramatic reduction in the cost. And then, of course, on top of that are the analysis costs, but that is steadily improving as well. And so that meant from a geneticist perspective that we are now moving to the situation where that genetic needle in the genomic haystack was a whole lot easier to find and has really been transforming the way we practice genetic medicine. And I'll give you some examples of that during the course of this presentation. Now, one example is an area that I've worked in for a number of years, the mitochondrial respiratory chain disorders. It's one of the most common of the inherited metabolic disorders, and the problem with this group of disorders is that the phenotype is incredibly broad, can mimic many different disorders, and the genetic diagnosis remains unknown for the majority of them. And we now know that at this point in time there are at least 250 genes that can cause a Mendelian mitochondrial respiratory chain disorder. And what this slide shows is the advent of various technologies in the identification of mitochondrial disorders. We have to remember that the first mitochondrial disorder was, or genetic defect was described in 1988 in the mitochondrial genome using a sequencing approach there. And with time, different approaches were used. But fast forward to 2010, And with the advent of next-generation sequencing, the number of genes identified through this technology dramatically increased, so much so that in the last 7 years, a third of all known mitochondrial disease genes have been identified, and that number is steadily increasing, as you can see there. I hesitate to talk about genomics and cancer, given Roger is in the audience, but I think it is fair to say that cancer is essentially a disease of the genome, and all cancers have at least a somatic mutation, so-called driver mutations that will push a growth advantage. And then there are other mutations, passenger or modifier mutations, that affect the pathogenesis or severity of the cancer in some way or another. There are, of course, many rare somatic cancers, and rare is defined by less than 6 individuals in 100,000 being affected. But when you lump them all together, they constitute about 20-odd% of all cancer. Germline cancers, so inherited cancers, represent about 5 to 10% of all cancers, and we know that there are over 300 inherited cancer syndromes. So as Roger very eloquently put, these omic technologies, including genomic technologies, have a great potential in the cancer space, both in terms of targeting therapy specific for the cancer, but equally in the germline cancers in terms of clarifying inheritance, penetrance, and risk for other family members, allowing appropriate genetic counselling and surveillance. So what potentially could genomic medicine, the introduction of next-generation sequencing into mainstream healthcare, mean in our context? Well, firstly, I think we can guarantee that there will be a faster diagnosis, which ultimately will translate to improved prognosis. As is very evident in the cancer space, and we think we're seeing in other diseases, we'll be able to be much more precise or targeted in the therapeutic approaches that we use. And indeed, we can introduce appropriate surveillance mechanisms for cancers and for complications of other disorders. Before I talk about the Australian Genomics Health Alliance, I think it's important to talk about the Melbourne Genomics Health Alliance, which started a few years ago and was the consequence of the coming together in a strongly collaborative manner of a number of the key research institutes within Melbourne who recognised that genomics needed to be developed in Australia, and so essentially this group, and who eventually got funding from the Victorian government, put together a statewide approach towards using omic technology, essentially prototyping the use of genomic technology for delivery of healthcare. And they were very keen to examine what worked or what might not work, what would work in a particular clinical context. And so their studies were a prospective study using whole exome sequencing and then targeting their analysis of that whole exome sequence data specific for the clinical phenotype that they were studying in terms of the various disorders. And this was carried out in parallel with the usual diagnostic paradigm so that the comparisons could be made. And so along the way, a number of shared approaches needed to be developed, including a single common consent form for all of these institutes in this space, development of standards of collection and cleaning up of the data, curation of the data, the various bioinformatics pipelines for interrogating the data and extracting what was relevant, and developing a common reporting format, and that was very largely informed by the establishment of multidisciplinary teams that reviewed the results and the clinical stories to ensure that what they were delivering in those reports were as accurate as possible and essentially clinical grade so that would be actionable in a clinical context. And perhaps one of the poster child for this study is the so-called childhood syndrome study. So this was a study where children who were recruited were under the age of 2, who had multiple congenital anomalies and facial dysmorphism, or had other features that people believe meant that there was a very strong chance that we were looking at a monogenic disorder, such as a neurometabolic disorder or a skeletal dysplasia. And so these children, again, underwent a traditional diagnostic approach, and parallel to that had whole exome sequencing. And this table summarizes the key results. Using whole exome sequencing, there was a five-fold increase in the amount or the rate of diagnosis compared to the traditional approach at about a quarter of the cost. And what surprised everyone was that in a third of cases, that new genetic knowledge translated to a specific intervention. So, and I'm not just talking here about improved genetic counselling. The new knowledge led to either a treatment being able to be instituted, or an appropriate surveillance approach being put in place, or inappropriate surveillance being withdrawn. And of course, one of the beauties of whole exome sequencing is the data is there, and you can come back and relook at it. And indeed, when the data was reanalyzed in the unsolved cases a year later, there were an additional 4 diagnoses made based on new gene discoveries that had happened in that intervening period. So this, of course, was very exciting and a really clear demonstration of the power of genomic technology. So enter now the Australian Genomics Health Alliance. This is a national health services research project funded by the NHMRC that commenced last year. And essentially it's built on the shoulders of the MGHA, aiming to develop an approach to delivering genomic testing at a diagnostic grade around the country. As you might imagine, there would be many potential hurdles or steps that need to be considered in that process. And in the next 10 minutes or so, I'll talk about some of the things that we're involved in making that happen. So, of course, in developing a national anything, there are all kinds of hurdles that one might face. First, of course, is when you're talking about delivery of healthcare, the divide between the states and the national healthcare systems potentially can lead to duplication of services between states, and at least in some instances can result in inequity of access to whatever the modality, diagnostic or treatment might be. On top of that, there is a lack of a capacity to store this large-scale data at a national level and to be able to share it. There's no common ethics approved processes for this at a national level, and many of our colleagues don't have much exposure to genomic medicine, and so there are educational gaps there. And so that poses the real risk of inactivity, fragmentation, and internal competition in trying to make this happen. On the other hand, one of the strengths that we do have is that by virtue of having a national health system, we can promote as best we can equity of access. We already have many NATA accredited laboratories that are working in the genomic space. There is a high level of genomic research happening in Australia, and we have fabulous clinical and research networks around the country that are just waiting to form these kind of alliances. And in addition to the national linkages, many of us have linkages internationally, and so the time is right for collaborating for this kind of approach. So the principle here is about moving the use of genomic technologies from a research setting where funding is unstable to the more stable situation where it's more or less routine and funding is sustainable through state and health systems. So we're talking about a whole of system change here. And there are many groups or craft groups that need to be involved in making this happen. And so the principal aim of the Australian Genomics Health Alliance is to demonstrate the cost effectiveness, the diagnostic efficacy, and the impact on clinical care of genomic medicine. This shows you the partners There are 60 partners nationally and internationally, over 200 investigators in this project, and at least some of the first part of last year was learning how to herd cats. But I have to say that it's been phenomenal, the level of interest and enthusiasm around the country in getting this to happen. And so much of the first year was involved in operationalizing all this. That's now happening, and we're now recruiting patients into the various programs. This gives you the structure of the AGHA, and there are essentially 4 programs. The first, program 1, is about building the clinical and diagnostic and research networks. The second is about big data. How do we collect it, clean it up, and federate it? The third is around the health economics and implementation science aspects of this project. And the fourth is around the preparing, or where are the gaps in knowledge, or where are the lack of individuals needed for the genomic workforce, and what are the social and ethical issues associated with this. And across those 4 programs, there are 2 major disease flagships that are being used to capture the data in real time, in real life, in the clinical setting. And so they're the rare disease flagships and the cancer flagships. So just to dive a little bit into the 4 programs. So program 1 is composed of expert clinicians and diagnostic laboratory people, coupled with research groups who have particular interest in the initial disorders that have been targeted for study. And the idea here is to essentially gather the evidence using these disease flagships to demonstrate the capability of this technology, and to garner the evidence to be able to demonstrate to state and health departments of health that this technology is really should be mainstream now and needs to be funded in a sustainable manner. A secondary aim of this, which is not really what AGHA is about, but which will be a potential beneficiary down the track, is that those patients who remain undiagnosed through this process will essentially, if you like, be enriched cohorts of patients for future gene discovery studies by research groups. 3 important initiatives of Program 1 are firstly trying to develop a standardized national consent for diagnostic genomic testing, and we have a working group on that at the moment. The second issue relates to variants of uncertain significance. So you'll do a genomic analysis, you may find variants in a gene that seems interesting or relevant to the clinical story, but it's unclear whether the particular variant in that gene is actually disease-causing or not. They're so-called variants of uncertain significance. And there's currently no strategy for being able to reinterrogate data and reclassify those variants in any kind of systematic manner. And that's an opportunity that's gone begging for a long time. And so we have a group working on what kind of processes could be put in place to be able to do this in a standardized way. And then finally, funding this kind of testing is, of course, a major issue. And so we have a group that's involved in harnessing the data that's being collected with a view to developing a rolling series of MSAC applications in the hope that this technology will be funded through the medical benefit schedule. Or there may be other funding mechanisms that are appropriate that involve Commonwealth and state partnerships. So Program 1 is piloting, developing recommendations directly within the clinical centres. So these are being clinician-led that will hopefully give the data that's needed to drive change in the health system. And as I mentioned, the 2 main disease flagships being used to garner this data are the rare diseases flagships and the cancer flagships that, again, are being clinically led. So the idea is that this is remaining clinically relevant. In terms of the rare disease flagships, these are the 5 sets of rare disease flagships that are currently in operation and recruiting patients. And later on this year, we hope to call for expressions of interest for a second tranche of rare disease flagships that would be funded for a 2-year period. In the cancer space, these are the three main projects that are running at the moment, and they're very much focused on the cancer surveillance and cancer therapy, incorporating both somatic cancers and also germline cancers, so trying to capture the various flavors involved. Program 2 is about big data. Again, there are 5 packets of activity there that we've identified as being important and needing to be standardized or harmonized at least. And the first relates around to just how people call variants and maintain consistency in reporting those variants. The second is capturing the clinical data and being able to match that up with the genetic data in as powerful a mechanism as possible so that you can make those subtle phenotype-genotype correlations, which will be very useful in terms of interpreting what we're seeing. And currently, the best-in-class software for bringing these two sets of data together is one called Patient Archive that was developed by Tudor Groza at the Gartner Institute, and we're in the process of linking that in with our program at the moment. Important, of course, is calling apples apples and oranges oranges, and so being able to develop standardized phenotypic nomenclature is critically important, and the standard in genomic parlance is the human phenotype ontology, but we've got senior representatives from CSIRO involved in this who have had a long-standing interest in standardization of phenotype nomenclature, and we're also very cognizant of the fact that electronic medical health records are on the rise, and they represent a perfect or potentially perfect source for being able to harness phenotypic data and link it with genomic data. And we're currently working with the Australian Digital Health Agency with a view to making that happen. And part of that will involve working with My Health Record. The fourth packet of work in this program, it revolves around the bioinformatics pipelines that are used to analyze the data. And this isn't about developing one pipeline to rule them all. Each centre has their own pipelines that they've developed over the years. And so what we think is important is to be able to compare the various pipelines in terms of performance metrics and so on, so that people can be confident about the use of those diagnostic pipelines in delivering genomic data. And then finally, and perhaps one of the most challenging, is how do we share data ethically at a national level and archive data so that it can be available for individuals to use in an ethically responsible manner. And that, of course, means we have to deal with state jurisdictions in terms of each state has their own privacy restrictions, and so this potentially is one of the biggest challenges we face. But the ultimate aim is that we will develop the strategy that we can present to government with regards to how can we federate these data and make it available in a re-identifiable manner, coupled with phenotypic data to be useful for clinical practice. Program 3 is about demonstrating to government that this technology is worth supporting, and so there's a very important health economics component to that in terms of capturing the data, being able to look at cost effectiveness modelling and so on. And the other string in that bow is determining what are the potential barriers to implementing this technology into clinical practice. And so there's a health and implementation science team that's involved in this as well. And finally, Program 4 is aimed at understanding what the education and training needs are for clinicians, not only specialist clinicians working in the genomic space, but all clinicians, because the reality is that we're all going to need to be involved in delivering this information to our patients if we're going to harness the maximum benefit of the technology. And so Program 4 is about identifying what the education needs are. It's not about developing the training programs, but it's about identifying where the gaps are and working with learned organisations such as the RACP to look at developing training modules. It's also looking at the psychosocial implications of genomic testing in terms of the impact on patients and families, and also examining a number of the ethical issues associated to sharing of this kind of information, uncertainty, incidental findings, and looking at different models of consent that could be potentially useful in a research and clinical setting. So it's important to acknowledge that Australia isn't doing this on their own. There are a number of major international efforts that are currently running, and the AGHA is very much closely embedded with those, particularly the Global Alliance for Genomic and Health and Global Genomics Medical Collaborative. So I guess finishing up, where are we in the Gartner hype scale with regards to genomic technologies? And people can perhaps put themselves in different places on this graph, but I'd like to think we're kind of somewhere around here, heading towards being able to make this a very productive technology that ultimately in the long run will make all our lives easier. And so finally, just to summarise the overall strategy of Australian Genomics, very much acting locally, targeting developments and embedding them within the different health systems at the clinician coalface, so that we can maximise our understanding of what will work and what won't work in real clinical practice. Building nationally, so I've talked about the Melbourne Genomics Health Alliance, but more recently the Queensland Genomic Health Alliance has formed and the New South Wales Genomics Collaborative has developed. So those two state governments are also have each poured in around 24 million each into looking at genomic technology and how it can be delivered. So the idea is working with those states, building nationally, we can develop a national network that will be most effective. And of course, linking internationally, picking the eyes out of what works in other countries and embedding that within our activities. We certainly don't want to be reinventing the wheel here. But I must say, one of the great advantages of Australia is that we are probably one of the few countries that can actually mount a national approach to this kind of technology. And even Genomics England is looking with interest at what we're doing, because they've found that they're struggling to get things to happen at a national level the way we think we'll be able to do it here in Australia. So on that note, there aren't any questions, but if you would like to find out more about the AGHA, the website is there, and we'd certainly be very happy to take correspondence from you. Thank you very much.